Hello, everybody. Welcome back to class. I feel like it's been a while since I've seen you. That's not the right word because I still haven't seen you. I think it's been a while since you've seen me. Last week, you guys did mostly your tests and stuff, so this week we're going to go ahead and get into the next chapter, get into the next lesson. This section is going to be on viruses and bacteria. I thought that might be appropriate considering what's going on in the world, that you guys understand a little bit more about how all that stuff works and the ideas behind it and stuff like that. We're not going to go into too much detail. We're just going to go into generally, what is a virus, what is a bacteria, how do they do their thing. Um, and this will be a pretty short section. We'll probably finish it. We'll probably be finished with it within the week. So let's get started. Let me close the door so my dog stop barking. I don't know if that helped. Okay. Viruses and bacteria. Here we see a picture of a bunch of what look like little blue robots attacking a gigantic hair follicle. But that's not what's actually happening here. These are viruses and they are attacking a bacteria. So viruses do attack bacteria, they do attack animals and plants and humans and all that stuff, but they can't attack bacteria. And this just kind of throwing you into it, letting you realize that viruses are way smaller than bacteria. So what is a virus? There's a little bit of a weird thing about viruses, because like we talk about killing the virus, different types of viruses, but you only kill things that are alive, and there's not really consensus on whether or not viruses are alive in the first place. You'll see the book introduces them as non-living particles called viruses, and most scientists do say they're non-living. They're non-living because they don't exhibit all of the criteria for life we see here. They don't carry out respiration, so they don't like breathe essentially. They don't really grow. They don't develop. They do make copies of themselves, so they do reproduce in a sense. They replicate themselves. But they can't do that without other things doing it for them essentially. They can't just sit there and like eat something and then do it. They have to make something else, make more of themselves. So it's a little weird. A cell in which a virus replicates is called a host cell. And what are they? Well, they are nucleic acids, so like DNA or RNA, enclosed in a protein coat and are smaller than bacteria. That's almost everything they are. Just a little piece of DNA or RNA, a little protein covering. Some of them have another little covering on the outside, and they're shaped a little bit differently, but that's all they are. We see down here they're given names based off of all sorts of things. They're not named in the same way that animals are named. Here we see a virus that infects a bacterium is called a bacteriophage, or just a phage for short. And that's what these are up here. I think those look the coolest, but you never know. Okay. Here's a 180,000 times magnification of what a virus looks like. That just gives you an example of how small these things are. So a virus has an inner core of nucleic acid, as we said, either RNA or DNA, and an outer protein coat called a capsid. Some big viruses have another coat, like I said, a little envelope, and it's pretty similar to your cell membrane. Not exactly the same thing, but pretty similar. But the fact that some of them have those membranes are why you can wash your hands and still destroy the bacteria, because you're still destroying that outer membrane. So then the virus can't do what it's supposed to be doing, what it wants to do. The nucleic acid contains the virus's genetic material. And again, it's either DNA or RNA, and it consists of instructions for making copies of itself, which is kind of strange. The only thing they do is make more copies of themselves. That's all they do, for the most part. They come in all sorts of different shapes and sizes. Here we see a couple of those, but the interesting thing is how they kind of attach to certain cells. So they're very specific. Many viruses are very specific in the fact that a certain virus will only affect a certain type of cell in a certain organism, usually. That's not always the case. It is possible for a virus to mutate and change targets, but most viruses are very specific in what they infect. And the way they do that is they have little connectors on their outsides, and these little connectors because the chemistry will just link onto special sites on top of cells. And when they do that, the virus almost gets activated, and then it starts its whole process of trying to replicate itself with that cell. So here we see an actual 65,000 times zoomed in picture. These little arms have met a specific little receptor on the cell. Now that little receptor, maybe that was originally for some like piece of food or a chemical or something, but the bacteria or the virus has a special little pretend hook on its outside that can attach on right there. So the cell thinks, oh cool, it's just a daily food delivery coming in, but no, it's actually a virus to pop in. This will infect the cell and in some cases kill it. As we can see here, there's a couple different shapes. We have polyhedral viruses, which look like, well, this looks like a D20 if you're a nerd, but different types of like dice and 3D shapes. I'm a nerd, that's why I know what that is. We got these cylinders, like the tobacco mosaic virus. Of course, they can always be spheres, which are just circles. And then they can look like those robots you see right there. Trying to look for a real-life example of one of these polyhedrals. Nope. Wow, who knew it would be this hard to find a dice? There we go. So viruses might look like this shape. This is a 20-sided die, but if you look at the way this is shaped, that is exactly the same shape as this guy right here. Each virus has a specifically shaped attachment protein. Therefore, each virus can usually only attach to a few kinds of cells. Oh, we've mentioned that already. And that's why usually if a certain virus affects a certain organism, we don't have to worry about it infecting another one. Unless certain mutations happen that allow the virus to essentially jump species, which can and does happen out in the real world. Here we see a little paragraph that says we have been able to eradicate a couple viruses because it was so specific that we were able to just stop it in its one spot, and now it's pretty much, smallpox is eradicated essentially. But things just like the flu, there's all sorts of different types of flu, and none of them are really specific as to what types of organisms they infect. So that's why the flu is such a big deal. I mean, aside from what's going on right now, just the regular flu never goes away because we haven't really figured out a way to stop it from traveling from species to species, from humans to humans, so it's a lot of stuff going on. So let's talk about how viruses actually replicate themselves. Once attached to the plasma membrane of a host cell, the virus enters the cell and takes over its metabolism. Remember, metabolism is just all of the cell chemical reactions happening, so it takes over all of the reactions. Only then can the virus replicate. Viruses have two ways of getting into the host cells. The virus may inject its nucleic acid into the host cell, like a syringe injects a vaccine into your arm. Another way the virus enters a host cell is after attaching, the plasma membrane of the host cell surrounds the virus and kind of sucks it up as if it were a piece of food or something that it was supposed to suck up. Those are the two ways. So it can be injected almost like a shot or like a vaccine that you normally get, or it can attach to the surface and pretend to kind of be something helpful, and then the cell will put a little bubble around it, suck it up, and then the bubble will pop once the virus is inside of the cell. Now there's two types of cycles that viruses can go through. There's a short-term one and a long-term one. Okay? This is the short term. And then lysogenic is the long term. So lytic is short term, lysogenic is long term. Short term, light term. Now some viruses do go through both of these. But go ahead and pause this video and watch the video I put in the link, and that video will help explain this with animations and fun pictures and colors and stuff. So go ahead and pause this and watch that video. Okay, this is assuming you watched that video. So you know about the lytic cycle and you know about the lysogenic cycle now. So if viral DNA that is integrated into the host cell chromosome, then that virus, that viral DNA, that virus is called a provirus. If it's integrated into the host cell's chromosome. It may not actually affect what the cell's doing normally. The cell's going to be able to keep doing what it's doing, but the instructions for the virus are still there. Which you saw in the video. That means once that cell can reproduce, it will hold on to some of those, some of that DNA. And as it replicates, all of the offspring of that cell always have that viral DNA inside of them. So these two cycles help explain why some things can show up again and again throughout your life if you're infected with them once, because the cells always have certain types of viral DNA in them. For example, the herpes simplex 1 virus, which is called a cold sore. So if you have a cold sore, you'll know that they show up and then they go away for a long time, and then they'll show up again and they'll go away. What's happening is once you get the cold sore, that's when the virus is in its lytic cycle, but when you don't have it, that's when the virus is in the lysogenic cycle. So it's still just building up until something like maybe extra sunlight or stress, whether that's physical or emotional stress. We're not quite sure what causes some of this stuff, but we do know that there is a triggering factor that causes these long-term dormant viruses to activate for a little while and go through their short-term. So that's why cold sores disappear and come back and then disappear and come back, because they're going through alternating cycles of this. Many disease-causing viruses have lysogenic cycles. Here we see the herpes simplex 1, which is just the cold sores. Herpes simplex 2, which causes genital herpes. Hepatitis B, that causes hepatitis B. Chickenpox, if you've ever had chickenpox, in some cases it can stay in there. And later in the life, sometimes for some reason, the chickenpox DNA that's inside of some of your cells will activate, and that will give you shingles. Hopefully that doesn't happen to anybody. Pretty rare, but I mean, it can happen. Now, when the virus gets released from these cells, remember the virus attaches to the cell, hijacks the interior machinery of the cell, has the cell make copies of the virus. The cell can spread the virus in two ways. One, it can make so many copies that it explodes, and that's called lysis. So the machinery inside of the cell makes so many copies of the virus that the cell explodes, releasing all the viruses everywhere. It can also just kind of it out right here. It goes in there and infects, it's in the DNA, and then it can make a little virus, and the virus can just go to the cell membrane and be popped out. Like if the cell was just, making one and then sending him outside, making one, sending him outside, without completely blowing himself up. There's just two ways the virus can get out of the cell, either by killing the cell or by becoming like a little package that the cell delivers. Here we see something called a retrovirus. Many viruses, such as the HIV, which causes AIDS, are RNA virus, which means RNA is the only thing that they have inside of their cells. Cells is not the right word. Viruses are not cells, so I should not have said that. Inside of the virus, essentially, there's only RNA. So once it's inside the cell, the virus has a little reverse transcriptase guy with him. That will help build the other half of the DNA. The DNA will then replicate and then throw that DNA into your cells. So now it's there for good. We can kind of detect if cells have these little reverse transcriptase guys in them. We can detect those, and that will let us know whether or not a person is infected with certain proviruses. Excuse me. So that little protein, that little enzyme, sometimes is the marker for us to be able to tell if a cell is infected or if a person is infected with certain types of viruses. The video went over this. with AIDS and white blood cells and HIV, so we are not going to go over this in this video. But you can imagine that if a virus is going into your DNA and kind of moving things around, putting its own instructions there, it might be possible that sometimes your DNA gets damaged in that instance. So we see in this paragraph, it's some viruses are linked to some cancers. And we can kind of make an educated guess that if you're in there messing with the DNA, it's possible that you damage something and now the cell is reproducing incorrectly. So that might cause some type of cancers to form. Besides viruses, there are also two other types of strange things out there called prions and viroids. Try saying those two really fast. Prions and viroids. Oh, that wasn't too hard. Prions are composed of proteins, but they don't have any nucleic acids. So they're essentially just little rogue proteins that go in, and we think they disrupt your body from making normal proteins by just kind of jamming themselves in there. And that can kind of cause things like mad cow disease. We got viroids, which are single circular strands of RNA with no protein coat. So that's strange if you think about it. Just a piece of RNA floating out there. They cause a lot of diseases in plants. And there's a lot less information on the RNA for the viroids than there are for viruses. And these are two actual pictures, 3,000 times and 6,000 times magnified. Little viruses infecting a white blood cell. Interestingly enough, the first viruses we found were in tobacco plants. These guys essentially filtered out what they thought was infecting the plants. Then they tried to filter that again the same way they filter bacteria and they kill bacteria, but it didn't work. So they realized this thing, whatever it was not a bacteria, and it was still able to infect things even after they essentially disinfected it from bacteria. So then they realized, hey, this isn't bacteria, this isn't the cell, this is something else that can infect things. And that's how we kind of found out about viruses. And then, again, plants always are kind of strange. We see here, this virus, this plant is infected by a virus, and all it's doing is making them have cool colors. So in that case, I mean, the virus isn't really, sure, go for it, virus, make the plants look nicer and make them more valuable. Like, it's strange. Plants always kind of weird compared to animals, but that's what they do. We see here, not all viral plant diseases are fatal or even harmful. So that's pretty interesting. Here we see the origin of viruses, and we're going to go over the theory of evolution a little later, but this kind of introduces you to a little bit. You might assume that viruses represent an ancestral form of life because of their relatively uncomplicated structure. Scientists probably don't think that. For replication, viruses need host cells already, so scientists suggested that viruses might have originated from their host cells. So some scientists think that viruses are like nucleic acids from cells that broke free from their host cells who knows how long time ago, and they somehow still have a protein coat to be able to infect people, infect other cells. So either way, they think cells are around and then viruses happened after, but we're not quite sure what the deal is here. And that's about it for section 18.1.